In microfluidics, generating uniform droplets is essential for applications like drug delivery, biochemical assays, and emulsions. But how are these tiny, consistent droplets formed? Scientists use three main methods: T junctions, flow focusing, and co flow techniques, each leveraging different fluid interactions. T junctions. Imagine a small crossroad where one fluid disperse phase enters from the side and meets another fluid continuous phase flowing forward. The interaction pinches off droplets at regular intervals. This method is simple, reliable, and widely used in emulsions and encapsulations. But how does the droplet size change? If we increase flow rate of the continuous phase, droplets become smaller because they get sheared off faster. Flow focusing here, the dispersed phase is squeezed through a narrow opening between two streams of the continuous phase, creating high shear forces that break the fluid into tiny droplets. This method is highly controlled and precise, making it perfect for biomedical applications like cell encapsulation. Co-flow techniques. In this setup, the dispersed phase flows through an inner capillary tube, surrounded by the continuous phase in an outer tube. As the fluids move together, the outer phase gradually stretches and breaks the inner fluid into droplets. This gentle method is ideal for producing stable emulsions where droplet size needs to remain uniform over time. Which technique is best? It depends on the application. T-junctions are great for simple droplet production. Flow focusing offers precise control for biotech uses, and CoFlow is excellent for stable, fine-tuned emulsions. With advances in digital microfluidics and AI-driven optimization, future droplet generation methods may become even more automated and versatile. Droplet microfluidics involves the formation of monodispersed droplets where immiscible fluids interact under controlled flow conditions. The generation of droplets depends on the interplay between viscous, interfacial, and inertial forces, which dictate the size, shape, and stability of the droplets. These precisely controlled droplets serve as microreactors for chemical and biological applications, including drug screening, single-cell analysis, and emulsification. Droplet-based digital PCR utilizes microfluidic generated droplets to isolate individual DNA molecules for precise amplification and quantification. Droplet formation in microfluidics is like creating uniform oil droplets in a salad dressing, where controlled mixing leads to consistent droplet size. The T-junction method generates droplets by intersecting two immiscible fluids at a perpendicular junction, where shear forces from the continuous phase pinch off droplets from the dispersed phase. The droplet size is controlled by the relative flow rates of the two phases, surface tension, and channel geometry. This technique is widely used due to its simplicity and ability to produce highly monodispersed droplets with tunable sizes. T-junction droplet generators are used in pharmaceutical microencapsulation, where precise control over droplet size ensures uniform drug release rates. This process works like squeezing toothpaste from a tube into a moving stream of water, where the water breaks the paste into small, uniform segments. Flow focusing designs use a central dispersed phase stream squeezed by two opposing streams of the continuous phase, creating a focused jet that breaks into droplets at a controlled pinch-off point. The droplet size is influenced by the balance between inertial and capillary forces, allowing for sub-micrometer precision in droplet generation. This method is particularly useful in high-throughput applications requiring rapid droplet formation with tight size distributions. Flow-focusing droplet generators are commonly used in emulsion-based drug delivery systems, ensuring homogeneity in encapsulated pharmaceutical formulations. Flow-focusing is like pushing a thin stream of paint through an airbrush, where the surrounding airflow precisely controls the droplet formation. The CoFlow technique involves a coaxial geometry where a central dispersed phase stream is surrounded by a continuous phase, leading to gentle droplet formation as the inner fluid exits into the outer stream. The droplet size is controlled by the relative flow rates and viscosities of the two phases, making this method ideal for encapsulating delicate biological materials. Unlike T-junction and flow focusing methods, CoFlow designs minimize shear stress, preserving the integrity of sensitive components. CoFlow droplet generators are used in cell encapsulation applications, where preserving cell viability is critical for tissue engineering and regenerative medicine. This method is like blowing soap bubbles through a thin straw, where the airstream gently forms bubbles without excessive force. Droplet formation in microfluidics is dictated by the competition between shear forces exerted by the continuous phase and interfacial tension of the dispersed phase. At high shear rates, smaller droplets form due to rapid pinch-off, whereas lower shear rates lead to larger droplets with slower detachment. Understanding these forces allows for fine-tuning of droplet size and stability in various applications, such as emulsification and microreactor systems. In cosmetic formulations, controlling shear forces in droplet-based emulsification ensures stable and uniform oil and water mixtures for creams and lotions. This principle is similar to breaking water droplets apart by stirring. A stronger stir creates smaller droplets. The ratio of dispersed to continuous phase flow rates directly influences droplet size, with higher flow ratios favoring larger droplets and lower ratios producing smaller ones. Additionally, microchannel geometry, such as constriction width and aspect ratio, modulates the droplet breakoff process, allowing precise customization of droplet size distributions. By optimizing these parameters, researchers can tailor droplet formation for specific applications. In food science, optimizing flow rates in microfluidic emulsification enables the production of stable nanoemulsions for flavor encapsulation. This process works like adjusting a faucet's water pressure to control the size of droplets forming at the nozzle. Scaling up droplet generation for industrial and clinical applications requires parallelization, where multiple droplet generators operate simultaneously within a microfluidic chip. This approach ensures high-throughput production while maintaining droplet uniformity, making it suitable for combinatorial drug screening and synthetic biology. The integration of automated control systems further enhances reproducibility and efficiency. High-quality put microfluidic droplet platforms are used in directed evolution experiments, screening thousands of biomolecular variants in parallel. This method is like using a multi-nozzle sprinkler system, where each nozzle generates identical droplets in high numbers. Emulsions generated in microfluidic devices require stabilization using surfactants, which prevent droplet coalescence by reducing interfacial tension. The choice of surfactant depends on the application, with biocompatible surfactants preferred for biomedical uses and fluorinated surfactants used in digital microfluidics. Proper stabilization enhances the shelf life and performance of microfluidic emulsions in diagnostics, drug delivery, and material synthesis. In lab-on-a-chip devices, stable emulsion droplets serve as reaction vessels for enzymatic assays, minimizing cross-contamination between samples. Emulsion stability in microfluidics is like adding soap to oil and water, preventing the two from separating over time. Beyond traditional methods, advanced techniques such as electrowetting, acoustic wave actuation, and magnetic control enable dynamic manipulation of droplets for smart microfluidic applications. These techniques allow for programmable droplet merging, splitting, and sorting, increasing the functionality of droplet-based assays in biomedical and industrial research. Future developments in digital microfluidics will enhance droplet programmability, paving the way for next-generation automated lab-on-a-chip systems. Electrowetting on dielectric EWOD technology enables precise control of individual droplets for automated sample preparation in molecular diagnostics. These methods function like touchscreen technology, allowing controlled movement of droplets with external stimuli like electric fields or sound waves.